Hello everyone, welcome back to another episode of STEM Nerds. I'm Jason. Thank you so much for being here in today's video. We're going to be talking about translation. That is right. We're going to be looking at how we take the messenger RNA that we learned about in the previous video. And if you haven't seen that, check that out. It's in the upper right hand corner. Really important to understand the process of transcription, what we talked about in the last video, so that we can now fully understand the process of translation, going from messenger RNA, translating that into proteins. So let's get started. And so we talked about this in the last video. This is the central dogma of biology. You have your DNA, and last video we explained that DNA as being like a cookbook. It holds all the recipes for all the meals / proteins a cell would ever need to make. But then when a cell is going to make a particular protein, it doesn't need the entire cookbook, and so it just needs a single recipe, and that is the messenger RNA. We talked about going from DNA to messenger RNA in the process of transcription in the last video. And now what we have to do is actually turn that code into something useful. Just like you wouldn't eat a cookbook, you wouldn't eat a recipe. Well, a cell really can't do much with the information until the information is converted into something useful, and that is the protein. That's kind of like the meal. The protein is going to do work inside the cell. It's going to have a function inside the cell. And so ultimately, going from DNA to RNA to protein, protein is our final, it's our goal. So how does a cell go from messenger RNA to protein in the process of translation? Well, that's what we're going to talk about here. Now, if you remember in the last video, this is kind of where we left off. We said that in a eukaryotic cell, the messenger RNA had to be modified. We called it post-transcriptional, after transcription, post-transcriptional modifications. And when last we left this process, we had messenger RNA that consisted of just exons, the regions of the gene that are going to be expressed to make a protein. We had our 5' cap, we had our poly A tail, and in a eukaryotic cell, the messenger RNA was out there in the cytoplasm, ready to be translated by ribosomes. And so for this process, we're going to need 3 different components. Our main players here are going to be the messenger RNA that we talked about in the last video. That is the code, that is the recipe. But we're also going to need transfer RNAs, otherwise known as tRNA. And the purpose of these transfer RNAs is to bring amino acids to the growing polypeptide chain, bring it to the ribosome, so that the ribosome on the right-hand side can actually read the code and put the amino acid sequences together to make our polypeptide or our protein. What's interesting is all 3 of these things are made of RNA. And so you've got the messenger RNA, you've got the transfer RNA, and then you have ribosomal RNA, which comprises the vast majority of the ribosome. So one of the things that we've discussed in the past is that this code is telling the ribosome what amino acids are going to be put in place when we're building this protein. Really, the specificity of our code comes from transfer RNAs. And what transfer RNAs do is they carry a specific amino acid, and that specific amino acid is based on what is known as the anticodon, that region down there in the purple that comprises the binding area, and that anticodon is going to bind to messenger RNA, and it's going to do so in sets of 3. And so the way that ribosomes read the code, read the messenger RNA, is by grouping the letters, the A's, T's, C's, and G's. In this case, A's, U's, C's, and G's. They group those codes in sets of 3, and that tells the ribosome, based on that set of 3, which amino acid is supposed to be inserted. And the specificity is really coming from the transfer RNA, because there's an anticodon there, and then we're just using our base pairing rules, A's and U's, C's and G's. And there's a temporary bond there, but that transfer RNA, in this case, that has AAG as an anticodon, is always going to carry the same amino acid, in this case, phenylalanine. And so that's where we get the specificity from. Now you're going to see there is some redundancy in the genetic code, and that is because we have to group them in sets of 3. If there were only 1 nucleotide per amino acid, well, there's only 4 unique nucleotides, and so therefore you'd only have 4 unique amino acids. Now you could pair them together, and if you did that, well, you would wind up with 16 potential combinations. That's still not enough to get us to the 20 unique amino acids that we need. So the solution is, if you have them in sets of 3, each position in that codon can give you 4 unique combinations, or 4 4 unique letters. 4 times 4 times 4 gives you 64. And so we have a lot more than we need, but we definitely have everything that is required. And so that's why you see some redundancy here. And so you'll see, as a really good example, proline. And so if you look at proline, it's in the second column there. Proline, there's actually 4 different codons for proline: CCU, CCC, CCA, and CCG. Well, that means any one of those 4 codons, if a ribosome recognizes any one of those 4 codons, it's going to insert proline in that position. Now, that third position has a unique name. It's called the wobble base. And the reason for that is sometimes it can change and you still get the same amino acid. We're going to talk more about this when we get into the different types of genetic mutations in a few videos from now. The interesting thing, though, is that the ribosome actually has a preference for one codon relative to the others. And so anytime there is a choice, it's almost like the ribosome has a preferred language. So for those of you who speak multiple languages, perhaps you have a language that you understand most fluently and you don't really have to think about it. You understand it right away. And if you're learning a language or you have a second language that you're not as fluent in, sometimes you have to stop and pause and think about what some of the words mean and think about what you want to say. Well, the ribosome is kind of the same thing. For the ribosome, it might recognize CCU as immediately like, oh, that's proline. And the other one, CCC, CCA, CCG, it might pause even for just a second. And even though it's going to eventually insert the same amino acid, what may happen is because the ribosome slows down the process of translation, the protein might fold ever so slightly differently. And what scientists have realized over the last 15 to 20 years or so is even though there might not be a mutation in the genetic code, and even though the protein might have the correct amino acid, sometimes the proteins can look a little bit different depending on the codon that was utilized to insert the amino acid. All right, so then you're going to notice we've got these 64 codons. The start codon actually has double duty. AUG, when a ribosome reads that, that is the place where it's using to start the process of translation or it's inserting methionine. And then there are 3 different stop codons. And so when a ribosome reads a stop codon, it knows, hey, this is the end of the messenger RNA sequence that I am supposed to be inserting amino acids, and it's going to stop the process there. So this is a very simplified way of explaining how the process of translation actually happens. And so we're going to start at the top, and you're going to notice that we have a ribosome. And it has 2 major subunits, the large and the small subunit. You're also going to notice that there are 3 locations within the ribosome labeled E, P, and A. And those letters stand for something. The A stands for the acceptor site. The letter P is the peptidyl site where peptide bonds are formed. And the letter E is for exit. And you're going to notice when the ribosome first attaches itself to the messenger RNA, the first transfer RNA that enters is going to enter in the P site. So that's at the very top of the screen. We have a transfer RNA that's recognized that AUG with it using the anticodon, and that's going to be methionine. Now the ribosome, what it's going to do is now the next transfer RNA is going to enter the A site, A for acceptor. And from that point on, the transfer RNA pathway is going to go from A. The ribosome will shift down 1 codon, in this case to the right. Whatever was in A will go to P. Whatever was in P will go to E. And when we get to E, we have an empty transfer RNA that is now going to leave the ribosome, E for exit. And so the first one entered P. The next one entered A. You can see the ribosome is then going to shift down 1 position to the right. And you can see all the transfer RNAs. Again, the order is we start in A. Transfer RNA will then go to P where the peptide bond will be formed. Transfer RNA now does not have an amino acid attached anymore. Will shift down to E, exit, and the ribosome will keep doing that, shifting down 1 codon to the right until it gets to a stop codon. And that stop codon, it doesn't code for an amino acid. So as that transfer RNA enters, there's nothing to attach. The ribosome disassociates, the protein disassociates from the ribosome as well. And so that's the end of translation. Now, if you remember, we've been talking about these whole 5', 3' numbers and how important they are. And the reason they're so important is really because of translation. And I know it's impossible to see in this picture, but trust me, if you were to look at the messenger RNA that's associated with this ribosome. On the left-hand side is the 5' end, and on the right-hand side is the 3' end. The ribosome, unlike the polymerases that we talked about earlier, the ribosome must start towards the 5' end and read the messenger RNA going from 5 to 3. It's opposite of the polymerases. This is why that directionality matters so much. Ultimately, it matters because what we need is an AUG start site that is the exact location away from or relative to the 5' cap, and that's where the ribosome starts. And so that 5' cap, remember what we said, 5' cap, at least in a eukaryotic cell, is helping the ribosome know where to start in conjunction with the proper AUG, because there could be multiple AUG codons for a single messenger RNA transcript. So two important things. The ribosome must read from 5 to 3. The ribosome is depending upon the 5' cap and the proper AUG start site to begin the process of translation. And so I thought this was a really cool picture here. You've got ribosomes that are reading messenger RNA, and again, they're just shifting down those sets of 3, the codons, 3 letters at a time, reading that. The transfer RNAs are coming, and you can kind of see them embedded here, which is really cool. And then as the ribosome is going from left to right, in this case, you see the protein coming out of the top of the ribosome. It gets to a stop codon, and that is the end of translation. Now, a few things I want to talk about, because there are some differences between eukaryotic and prokaryotic transcription and translation. We mentioned a few in the last video, but here is a really good illustration. What we have here is transcription and translation of a prokaryotic cell. Remember, prokaryotes, they don't have nucleus. They don't have a nucleus. They don't have membrane-bound organelles. And so what winds up happening is you can see that RNA polymerase is reading the DNA, but before the RNA polymerase is even finished with transcription, you can see ribosomes begin the process of translation. And this is possible because we don't have any introns. There's no such thing as an exon. Every part of the messenger RNA for a prokaryotic cell is translatable. There is no nucleus that is separating the ribosomes from the messenger RNA. There's no caps, no tails. And so what we have here is RNA polymerase going through the process of transcription. Before that finishes, ribosomes begin translation by reading the proper AUG start site. And you can see here, the two can happen simultaneously, and it actually speeds up the reaction time for these prokaryotic organisms. All right, so then if we were to summarize the differences between eukaryotic and prokaryotic transcription and translation, again, just like we just said, in prokaryotes, transcription and translation can happen simultaneously. That is because we don't have to worry about introns, caps, tails, or a nuclear membrane separating the RNA from a ribosome. Again, no introns, no exons, no caps, no tails. And the other thing, and this is kind of a newer concept, in prokaryotic cells, they are polycistronic versus in eukaryotic cells, they are monocistronic. And what that means is in a single genome or in a single transcript, you may have multiple genes and multiple proteins coming from a single transcript in prokaryotic cells. In eukaryotic cells, that is highly unusual. Usually a single messenger RNA transcript is for a single gene. And so to summarize, polycistronic in prokaryotic cells means a single transcript might hold information for multiple proteins. In eukaryotes, being monocistronic, a single messenger RNA transcript will probably hold information for just one protein. All right, so that was a brief overview of translation. In the next video, we have to talk about how this stuff is controlled, because we can't just have transcription, translation happening all the time. Cells need to be able to turn genes on and turn them off. That's what we're doing in the next video. As always, thank you so much for being here. Have a great week, everyone.